next Wednesday, I'll be able to give you a very clear statement on where we stand with the textbook. I'm aggravated because I switched textbooks because we couldn't get the first textbook. And it just brings home again the problem that we have to write a new textbook in this field. We're very short on available text. Today, as I promised you, and you'll have to keep the noise down, folks, it's a big room. I let all you in with the understanding you can be quiet. Remember the deal we made? Right? Extreme quiet, because there's lots of people in this room. Today we have a special treat. As I said, almost every year that I've taught this, I've offered a videotape of Professor Colin Pittendrigh of Stanford University, who gave an introductory lecture on the history of biological timing. And this year we are extremely fortunate. Dr. Pittendrigh is actually visiting the University of Virginia and has graciously agreed, even though he's still suffering from jet lag, he just got here very, very recently, he's agreed to give us a lecture on the history of biological timing. It's a great honor to have him here, and I'd like to turn over the lecture to him now.
Thank you. Is the textbook the handbook of... I exhort you all to buy it. Anybody who buys 5 copies gets A. 4 copies B. I get royalties. Well, I'm going to attempt to give you some sketch of the history of the field, but not so much, I'm not going to labor historical details as to try and sketch for you in this hour what the field's all about and how it came to be in recent times. There is some history to it, to be sure. The first recorded observation that's relevant to what you're going to be looking at was made as far back as 1729 by a French, we would say today, earth scientist, a sort of geologist, a gentleman by the name of de Mairan. And Monsieur de Mairan, as he referred to himself in publication in the third person, became interested, as many other people had, in the fact that the primary leaves of a bean seedling, the primary leaves, by day are up, horizontal, and at night they come down into a vertical position. And there is a regular daily movement of the leaves up and down, horizontal by day, vertical by night. De Mairan asked himself, what causes that movement? And in trying to answer the question, he put the plant into what he calls a dark chamber, expecting that the rhythmic movement of the leaves would stop. It did not. It persisted, to his surprise, for several days. It's of interest that de Mairan himself, 1729 please, recognized this as a very important observation, sufficiently important that although he was a geologist, he reported it professionally into the Transactions of the French Academy. It's there for us to read now. And in that brief note, he says some wonderful things. He says, this is important, but I'm busy with other things. And he recommended it to his colleagues, the biologists, the botanists, and the physicians. Then he says, progress will be slow. You don't get that. Progress will be slow. He's turned it over to the botanists and the physicians. Yes. But actually, that isn't why he predicted it would be slow. And there's a point in my going into this detail. It was slow. It was essentially 200 years before any progress was made. He says in his note, progress will be slow because true science, I won't risk the French, true science, which is the experimental kind, please note 1729, is necessarily slow. What that all means is that de Mairan didn't see what the next experiment was. He didn't know where to go from there. Now, while we're at it, asking questions in science almost always amounts to doing an experiment. And when you're doing an experiment, you're asking a question, how nature works. And Demiran didn't see what the next experiment was. He didn't even know what the next question was. There is a point here that sometimes observations are made which science cannot handle. Let's notice that. Sometimes it's too soon to talk about something. It's too soon to talk about consciousness. It's outside the domain of science. Nobody knows what the devil consciousness is. No grams, centimeters, no seconds. Consciousness, forget it. It hasn't entered science. And in 1729, this observation didn't seem to be tractable by the discussion of nature, which is science. 200 years elapsed. And during those 200 years, it wasn't that Demiran's observation was not known. It was known to every major botanist in Europe. We won't bother with their names. They wouldn't mean anything to you. Except, for instance, Charles Darwin was aware of Demiran's observation. And it's not unimportant, we're not going into the details, that he made the best noises about Demiran's observation. But nobody really got anywhere. The name Pfeffer, P-F-E-F-F-E-R, won't mean much to many of you at this time, but Pfeffer, often described as the father of modern plant physiology, the person who discovered osmosis. Most of you will know what osmosis is. Pfeffer was fascinated by Demiran and wrote 2 big books about Demiran and experiments. But it was not until the 1930s, 200 years later. Pfeffer, by the way, is about 1910. Give or take 10 years. One book 1890 and the other one 1920. So 1930 there was some kind of a break. But before we go to what that break was, why did it go so slowly? There were 3 elements about Demiran's observation and related phenomena. Many other observations had been now made, similar to Demiran. Have you ever heard of click beetles? When click beetles click and come off if they're on their backs. And if you put them in a tin and they click, of course you can hear them. Get a marvelous Demiran rhythm of click beetles. If you put them in a tin, there's a periodicity to the clicks. All kinds of rhythmicity had been observed by the 1930s. But the three features of the discussion which were profitless. Please notice sometimes the discussion of nature is profitless because you're not asking useful questions that clarify what's going on. The 3 features of the early discussion were: 1, there was it memory? Was the plant remembering the experience of 24-hour cycles of light and darkness, 24-hour cycles of temperature? Was it a memory phenomenon? Or was it inherited? Was it innate, built in? That discussion back and forward, both possibilities in the literature, but nobody clear. The second thing was that appeared when a Swiss lady, Rosa Stoppel, S-T-O-P-P-E-L, Stoppel, published the proposition that these rhythms that persisted in constant darkness in the laboratory were really being driven, caused, not by the light-dark cycle, which you can get rid of, but by some unknown factor X, something associated with the Earth's rotation. Some geophysical cycle was driving the rhythm. Well, you know, any hypothesis that's not testable is not part of science. But actually, Stoppel's is testable. And in Germany, a botanist, Kremer, C-R-E-M-E-R, took Demiran's plants, I mean new ones, but 200 years later, but the same species, took it 600 meters down a salt mine in Westphalia and reported, aha, the rhythm stops at 600 meters down, factor X, Stoppel's factor X it was called, this mysterious periodic variable that's due to the Earth's rotation. Whatever it is, factor X, it's screened out at the bottom of the salt mine. Marvelous, and the rhythm starts when you come back to the surface. Nobody's ever repeated that, tried, and we don't know what misled Dr. Kramer, but something did. Other people have taken other rhythmic systems down the same salt mine. They persist. But I'm telling you, there was a lot of whatever distraction from the main issues. Memory, heredity, factor X, or built in, not clear. For my own part, I also believe that the third feature of the 200 years of fruitless discussion was that nobody saw any utility or function, as one would say, to this sleep movement of the leaves. It's an interesting fact to this day. The botanist can't really tell you what the plant is buying when it moves its leaves up and down. What's the utility? What's the function? And I stress this for two reasons. That asking what the function is a very important feature of the biologist's discourse that the physicist never has to ask. You don't ask what the function of electronic orbitals is, what good it does the atom. But you ask what is the function of a digestive system, a respiratory system, what have you. What is the function of this spectacular periodicity which persists in constant darkness? Another little side note, we're going to get to some biology in a minute, that amuses me, more than amuses me. Science itself, which is looking for a language to discuss experience, to discuss nature. That's somebody's one definition of science, and it's a good one. Evolving a language for the useful discussion of what we observe. Evolves. And there's something about the progress in science which is a little bit analogous to the way organisms evolve. It isn't simple growth. Critical opportunities arise in the way people think about things that transform it all, and all of a sudden it explodes. Well, the absence of any discussion or insight as to what the utility of de Mairan's rhythms was, and I close this discussion, this part of the discussion there, was, in my view, a major feature in why it took 200 years to take off. Now, in the 1930s, there was a series of papers by two Europeans, both Germans, a man called Hans Kalmus and another one, Erwin Bühning. And both of them published papers which were of great importance in the history of field, and in more than one way, as you're going to see. One I'll tell you about very briefly, but both of them used a rhythm that persisted in constant darkness like this. And each one of these things here is an event that happens once every about 24 hours. Now I gather, before we go on, so I don't put this monster on, that you've seen this, have they already? That happens to be a figure of mine, as a matter of fact. And what it's telling you is you see all these individual black lines. Those bands of black markings are from a recorder indicating when a mouse, a field mouse, is active in its cage. It gets onto a running wheel and runs. And when the running wheel is moving, you get a record of it. And you can see that each line, which is a successive day, up on the top, you see two vertical lines, which are lights. It's a light, an hour light each day. And the light comes on once every 24 hours. And the mouse is active once every 24 hours when the light goes out. When you turn the light out, oh yeah, I can do that here, can't I? When the light gets turned out here, oh, thanks, terrific. When the light gets turned out here, they're in constant darkness, and the animal's rhythmic behavior persists, but the onset of running wheel activity gets earlier each day. And if you fit a line to those onsets, you can measure the period, which in this mouse was about 23 and I don't know how many minutes, 23 and a half, let's say. And then down here, light is turned on again, and the light-dark cycle is regulating the rhythm to exactly 24 hours. The fact, this is Demiran, the rhythm persisting in constant darkness without external cues from light or temperature. And I'm sure you know by now that in the 1960s, the word circadian, which has the roots circa, about, Latin, and dian, days, it's a rhythm the period of which is about a day. We're talking about circadian rhythms, which were discovered by Demiran. Well, here's a circadian rhythm, and it happens to be something that the fruit fly Drosophila does. And we're not going to, at the moment, bother with the detail of what it is. It happens once a day. And when the insects are released into constant darkness like that mouse, you get a burst of activity once a day in constant darkness. The important experiment that Hans Kalmus did was to take these insects, they're actually the pupae, not the adult flies. He took the pupae and put them into pure helium, no oxygen. No oxygen, no oxidative phosphorus, no respiration. Respiration stops. The insect's energy supply to drive anything to do with its metabolic activity stopped. And the result was, if that's the control, that this peak that is expected here is delayed the same amount of time as the helium. In other words, if the helium's on for 6 hours, this peak's 6 hours late. Beautiful. Rosa Stoppels had it. If it were factor X, some mysterious external cycle that's driving the rhythm, that cycle doesn't care whether the insect's on the verge of death from anoxia. Keeps going. But that's not what happens. If you stop metabolism, you stop the rhythm. A very beautiful and important result. Bunning and Kalmus both did a comparable experiment in which, instead of using anoxia, one says, stopping the rhythm by withholding oxygen, instead of doing that, they dropped the temperature here from 26 degrees temperature to 16. Now, what's the point of this experiment? Again, 1930, everybody still has to worry about Rosa Stoppel. If Stoppel is correct, the temperature doesn't mean anything, have any effect on the rhythm, because some external cycle that's independent of temperature is driving it. If, however, it's of metabolic origin, and this experiment was done before the helium one. If the rhythm has a metabolic basis, the period of the rhythm should be temperature dependent. The rhythm should go faster at higher temperatures, slower at low temperatures. And both of these men, with slightly different designs to their experiment, dropped the temperature 10 degrees and found that this peak was delayed about 12 hours. Stoppels had it again, that's fine. This experiment is completely misleading. I'm going to come back to, but that's part of the fun. But the helium experiment in itself leaves no doubt that the rhythms of internal origin and that its motion is dependent on metabolic energy. Now, you still have the question, is it a memory phenomenon or is it built in? Bunning answered that in a beautifully simple experiment, again with fruit flies. He and others before him, we don't need names, they're unimportant. That's not fair to the people, but it's not important to the story. And if you want him, a man called Bremer, never mind, found out that this Drosophila rhythm, like many other circadian rhythms, but not all, stopped completely in constant light if the light intensity is high enough. In fact, the rhythm is wiped out in constant light. So what Bunning did was to put Drosophila, easy to culture, into constant light for 30 generations. Drosophila melanogaster, about a year we're talking about. No rhythmicity in the insects at all. And one speaks of constant light, there's jargon in the field, light, light, continuous light, stops the rhythm. Then he transferred the insects from constant light to constant darkness, jargon D, D, and immediately the rhythm is initiated. These insects and their parents and their ancestors haven't experienced 24-hour periodicity for 30 generations. But as soon as the light is turned out, they spontaneously enter into a circadian period. So the system is innate, it's inherited, it's not memory. And Bunning did other things. He had 2 strains of beans, which we've talked about. And these 2 strains differed in the exact period of the rhythm. And he crossed the 2 strains, and the F1 generation was intermediate. And those of you who know some genetics, the F2 showed the big spread. Genetic control of the period of the rhythm. 1930. The whole field could well have taken off from there. World War II came, and all that kind of science stopped. And I'm not sure whether the foundations were really still there. Look, I want to come back to this. For 200 years, the sleep movements, as they're called, sleep movements, the leaves go to sleep, it is said, when they drop. The sleep movements, no known function. Don't know what the plant's buying with it. But that was the experimental system, one says, and everybody used it. So, and this is what's called, this is a historical accident. It's the kind of thing that happens in the history of life. The field was founded using a system in which nobody had any guess that there even was a function. So functional questions were never raised. And if this were a textbook account, one would have to be fair and say that Bunning in 1936, same Bunning, introduced one element of function, which I'm going to come back to. But it certainly didn't have time to take off, the idea, before the war interrupted it all. And in my view, this is a personal view, the field really took shape and became interesting in following the year 1950. And I'm going to tell you, because I think you'll enjoy them, the experiments, or some of them, that transformed the field. In a history of my own interest in this, I happened to hear a lecture by one of the 2 schools, a man called Gustav Kramer, who was killed, fell off a cliff in the course of a later experiment in the late 50s. Kramer and the great Austrian naturalist Carl von Frisch, that many of you will have heard of, von Frisch, student of honeybees. But Kramer did the better job. When I heard his lecture, I was overwhelmed. I hadn't read the literature, which was only 6 months old anyway. And his lecture was one of the most marvelous experiences I've ever gone through. And it set me off thinking. What was the story? Cramer himself, not at all interested, not professionally committed to bird navigation, migration, was one September afternoon walking through the grounds of his institute, which was in the German town of Wilhelmshaven, North Germany. And he noticed that about 50 or so starlings, these exotic birds, starlings, that one of his colleagues worked with in a big outdoor aviary, the aviary consisting of chicken wire, holding the birds in. He noticed that all the birds that afternoon were clustered, trying to get out to the southwest. He knew that at that time of the year, all of the North European starlings were in migration, headed for the winter in Spain, to oversimplify, southwest. And it occurred to him these birds were trying to navigate. So here, you know, biologists are always looking for an experimental system, something you can do experiments on. Nobody knows how birds find their way. What about this? So he built, and I have to be quick here to get through other things. He built a device inside of a hemispherical tent. And he made use of a discovery that a Finnish biologist, Palmgren, doesn't matter about names, had discovered and published during the war years. Palmgren discovered that if you put a bird in a cage, doesn't have to be gilded, put the bird in the cage and put it on a perch. That one went down marvelously, right? Put it on a perch that rotates. During the breeding season, the bird will settle on the perch and head in the direction that it would fly were it free. That was Palmgren's observation. So what Cramer did was to take a starling, put it on such a perch, which was rotatable, inside a tent. The tent had doors to it. It's a pretty elaborate tent. You could open windows. And Cramer discovered that if you put the bird inside the tent so it couldn't see the sun, oh, I've missed a point. Before he got to his tent, we're going to use it in a minute. He noticed that afternoon, watching this, that all of a sudden the birds would become random in their distribution. And then they'd also go back to the southwest. And it only took 2, 3, 4 such incidents to notice that the birds headed southwest when they could see the sun. But when the sun was clouded over, they went in all which ways, all directions, random. And you come to the remarkable conclusion, if you're Cramer, that the bird is using the sun in some way to tell it where the southwest is as a direction giver. And I'm told, I haven't seen it, that there is a correspondence between Cramer and another great German biologist, ornithologist, Stresemann. And Cramer, I'm told, is said to have written to Stresemann, I can't believe it. But it looks as though the birds are using the sun. So he built this tent, I told you, and found that when the bird is put in the tent and the windows are closed, just like clouding over, the bird settles on its perch in all random directions. Open a window such that the bird can see the sun, and it immediately heads southwest. I don't know how immediately, I haven't done the experiment, but it heads southwest. Lovely experiment. Open a window with a mirror on the inside. So what the bird sees is not the real sun, but a virtual image of the sun, and it makes a 180-degree error. Lovely. So now, he's really encouraged that the bird is using the sun as a compass. And everything's going well when the migratory season's ended. Birds uninterested in the southwest. And another letter to Stresemann, so I'm told, I haven't seen them. Someone called Ernst Mayr has told me about these letters. he says to Stresemann, I can't wait for next year. You can't wait a year for the damn birds to become interested in the Southwest again.